Okay, so welcome back. This is a change of scenery. We're still on the general theme of life is defiance, but now we're going to move to a different example. So, in the previous three or four vignettes, we focused on the question of the proton waterfall or the concentration gradient waterfall. And that's a beautiful image. It's got an associated set of really fun physics questions and some fun sort of mathematical treatments, and I gave you my impressions of how to think about that in a sort of elementary fashion. And now what we're going to do is we're going to tackle the one in the upper right. So I want to point out now that adaptation is another really key biological process, and it's a biological process that requires the investment of free energy. And so I want to use the example of bacterial chemotaxis as our first entry into this general class of defiances. So let me just go back more than 100 years, let's say to the 1890s, and we're talking about experiments of Engelmann, who, these kinds of experiments are a testimony to the cleverness of people. So this is in principle an experiment even that Newton could have done. And what I mean by that is, we're living through the pandemic right now. It's 2021, February. In 1665 and 1666, Newton was banished from the University of Cambridge. He went back to Woolsthorpe, which was this manor near Grantham in England. And while he was there, he worked on the inverse square law. And I think he understood at that point that the orbits that resulted are elliptical. He invented the calculus and figured out the laws of motion as we, roughly know them now. And he invented the reflecting telescope. And finally, he also did really interesting optical experiments. And there's some cool drawings. I've actually been in the room where he did them, where he brought light through a pinhole basically, and then passed it through a prism. It's separated into its spectral colors. And then he asked himself the question, which the answer to, but he didn't. And it's an experimental question, really. Once you understand it, of course, it's easy. But he didn't, and so he passed light through a second prism. He'd select, for example, the blue or the green wavelength, and then he would pass it through a second prism in order to see if it would disperse again, which it didn't. At any rate, what's shown here in Engelmann's experiment is that you have light that is separated spectrally, and then you have cells that are living on the cover slip, which is exposed to these different wavelengths, and you watch in a microscope. And the outcome of that is shown here. So I always loved this. I think Julius Adler is the first one who showed me this, and this is the kind of thing that really moves me, because there's an elegance and a simplicity to the experiment. There's a crystal clear statement of what the outcome is. We could call it phototaxis, but in fact it has to do with gases and things like that too, if I remember correctly. But the upshot is that the microbes express preferences. What I mean by that is that they care where they are in space because of the changes, the heterogeneity in environmental conditions. So that's the backdrop to the general subject of axis. It could be phototaxis or chemotaxis or galvanotaxis or whatever. And these different kinds of taxises reflect the fact that living organisms know how to respond to their environments, and that's borderline triviality. So now what I want to do is I want to turn to the subject of chemotaxis. So this is an image that was taken by the great Julius Adler, who you could read a very nice retrospective that he wrote in one of the annual reviews journals. Julius Adler was an extremely clever experimenter in the early days of chemotaxis, doing assays in which he precisely quantified the number of cells as a function of the concentration of chemoattractant inside of his small pipette. So what you're seeing here is tons and tons of cells which are collecting at the entry point to a pipette. The pipette has got some chemoattractant in it, and what he is imagining is that there's this gradient of chemoattractants, and the cells are attracted to that. And we can mathematicize all of these sorts of sensibilities, and this is the sort of thing that people like Howard Berg has done with Victor Surik, have done really beautifully, and in fact that's going to be the jumping off point. for what I want to talk about. So bacterial chemotaxis is a really interesting process that involves a combination of what are known as runs and tumbles. So, here's my bacterium, and it will undergo some long, let's call it a 30 micron long trajectory. It will reorient, and then it'll go off, and reorient again, and then it might go off in this direction, and so on. And so the point is that this part is known as a run, and this part is known as a tumble. And the concept is that there's, let's say on the order of 5 flagella, which you can see very evidently up here. They've been fluorescently labeled. So this is from a paper of Turner and Howard Berg. And when the motors, the rotary motors that turn the flagella, when they change the direction of rotation, it causes the flagella to splay apart. The bacterium will reorient, and then it will start the motors in the same direction again, the previous direction, at which time the flagella will act collectively, and they will lead to one of these run behaviors. So that's awesome. This whole thing is mediated by a relatively simple but also relatively complex circuit. I think it depends on what point of view you come from. If you come from eukaryotic biology, you're going to think of this as an extremely simple circuit. And if you come from condensed matter physics, you might think of this as a really complicated story. And let me try to just give you some of the essences of it. So, essence number one is that these chemotaxis receptors are pointing out into the world on the lookout for chemoattractants, which I've labeled here circles, squares, and triangles. When one of these chemoattractants binds to its cognate receptor, that receptor will actually perform a phosphorylation reaction on a signaling molecule, and that will tell the motor basically to keep on running, if you like. Now, if there is an absence of signal, then the motor, the cell will think, there's no reason to, I'm not improving my position, I'm not improving my direction of swimming, and so why don't I reorient? So what I'm trying to tell you is that this motor, which turns the flagellum, it rotates in 2 directions. It rotates in one direction, which leads to forward motion, and it rotates in another direction, which leads to splaying the flagella, and thereby the rotational reorientation of the cells. And the equipment, the essential equipment, is the receptor, shown here, the signaling molecule, known as QY, and then the motor. That's the real essence of it. So if you want to describe this simply to somebody at dinner, you would not talk about all of this extra business, that I'm for the moment not mentioning, but I'm going to mention. And you would just say, yeah, there's a detector, and then there's a messenger. That's the essence of it. These are the detectors. This is the messenger. The messenger carries the message of whether or not there's good stuff around, and that leads to changes in the behavior of the motor. All of this extra stuff is the equipment of methylation. Methylation is the mechanism of adaptation. So, what I'm telling you is that this receptor can be decorated with up to 4 different methyl groups. Again, the specific details of this I don't want you to get tripped up on. Of course, they're super interesting, and the experimental achievements and also the intellectual achievements of those who figured it out are to be absolutely saluted. It's really, really awesome. I don't question that for a moment. But what I'm saying is that let's try to boil off all the sort of notational nomenclature complexity and focus on what is. And for me, what is that there's a detection system, which is, as is often the case, mediated by membrane-bound receptors. These receptors, beautifully, can exist in both an inactive and an active state, and we're going to have a lot to say about that, but I've already hinted at that several times, and I would call that one of the great themes of the course, and one of the great themes of science, actually, is this sort of, it's in a way the John Tukey bit, binary digit mentality, that somehow zeros and ones allow us to say a lot about the way the world is. that we can capture the essences of things. And so this sort of binary yes, no, on, off, active, inactive concept for proteins is a very powerful one. So these receptors can either be on or off, and if they're on, then they will put a phosphate group on this key Y, and that changes the behavior of the messenger and tells it, go do your job. That's the essence of the thing, which then leads to reorientation of the motor. And all I'm trying to say is that then that gets modified, and one of the beautiful ways that it gets modified is that you can change the sweet spot. And what I mean by that is that normally the receptor is living in such a way that there's a midpoint to its activity. And that's based on what the ambient concentration is. It's trying to detect gradients. But if I shift the overall absolute concentration, these systems are smart enough, with quotes, they're smart enough to change their midpoint. That's the part that's awesome. That's not part of the Langmuir binding worldview. So, what we're going to do here is we're going to defy Boltzmann and Langmuir. So let me just tell you about this adaptation. So what I'm showing you here is, let's call this first frame. This is homeostasis. There's some background chemoattractant concentration, C1. And the system, if you'll let me use these words, is tuned so that its midpoint is exactly where it will be most sensitive to changes in concentration. And it's got a run and tumble profile like I'm showing you down here. So in other words, these flat regions are the run, and then this little dip is the tumbling time, and it keeps on doing that. So as you can see, this period of time, the flagella are splayed out. During these long episodes, the flagella are bundled, and this thing is moving along. Now what I'm saying is, let's imagine a concentration jump. So at this moment, right here, we've gone from this state to this state just by changing the concentration in the medium. We pipetted in concentration C2. Now the cell detects that higher concentration, and it's living over here on its curve. And so it thinks, oh my gosh, I'm moving in the right direction because now I'm going towards higher concentrations. But in fact, we tricked the cell. And when I say we tricked it, what I mean is that we persuaded it that there was a gradient when in fact there's not a gradient. There was a gradient in time, not in space. And so after a little while, the cell's going to say, oh, you know what? The concentration is uniform. I'm interested in detecting changes, and so I'm going to redefine my midpoint such that I'm once again in the sensitive part of the curve. And that's the adaptation. And the way that happens is by tuning some charged groups, methyl groups that are put on the receptor, which changes its activity. So that's the real exciting point for this particular part of the course, which is about adaptation as a kind of defiance, a defiance of standard Boltzmann binding. Boltzmann binding says, I have a receptor, there are ligands around. The ligand receptor binding takes place according to the states and weights that I did earlier in the term. And your midpoint is your midpoint. There's not anything you can do about that. Here, we're gonna put together the MWC model, which tells us how to get an activity curve like this, which is sigmoidal and cool. I love it. And then we're going to include above and beyond that changes to the MWC framework as a result of these methylation events. So I think that's the background that I wanted to give. So what have I done? What's the summary? What's the framing? The framing is that, first of all, we're talking about the part of the course that has to do with adaptation, now, which is part of the more general theme of defiance. And that's great. And I'm trying to tell you about a whole suite of different experiments which have to do with the so-called axises, chemotaxis, phototaxis, galvanotaxis, etc., again, durotaxis, where cells are responding to variability in their environment. And these experiments the classic experiments from Engelmann are wonderful. The classic experiments from Julius Adler are wonderful. And then the sorts of things that Howard Berg and Victor Surek have done at a much more sort of microscopic and fine-grained level by using FRET to examine the activity of the messenger molecule, for example, or the receptors. And then examining the meaning of this adaptation is going to be just a really wonderful part of what we talk about. So that was basically embodied in my little narrative at the end about shifting the midpoint. So that's the thing I want you to carry away from this. It's something that you're used to. You shift the midpoint every time you get up in the morning or go to bed or whatever. If you especially after you go to bed, if you try to look around, you'll find that you're not doing a good job of being able to see. But if you wake up in the middle of the night, you can walk around, and that has to do with adaptation in your visual system, which takes place at the level of not only of your diameter of your pupils, but also at the level of the molecules themselves. So anyway, this sets the stage for the things that we're going to do next.